project Hexatry, which is focused on the bioremediation of chromium-6 contaminated wastewater. The start of our project began when we read in the local news that there were elevated levels of chromium-6 in drinking water. And this really posed, we were kind of alarmed that this would pose a threat to human health in our community. And so we were very intrigued by this chemical species, so we sought to find out more. So chromium-6 is a toxic carcinogenic heavy metal chemical species, and it can be reduced to the oxidation state chromium-3, which is relatively non-toxic, and it actually precipitates out of water. So we plan to use these properties to address the issues surrounding chromium-6, as our wet lab team will elaborate on later. And we also wanted to find out more context about what existing laws and regulations affect the actual levels of chromium-6 in our drinking water. So we found that at the federal level, there is a maximum contaminant level of 100 parts per billion of total chromium, which includes both the trivalent and hexavalent oxidation states. And however, there's a big discrepancy with the California public health goal. With the California public health goal that recommends a maximum contaminant level of 0.02 parts per billion of chromium-6 specifically. And this level correlates with correlates with the level in literature that suggests that at this concentration, there's an uptick in cancer rates as a direct result from consuming chromium-6 contaminated drinking water. So given this regulatory context, how are we actually doing in terms of chromium concentration? So we found that at the national average, it's at 1.04 parts per billion of chromium-6. And since we're interested in Houston specifically, we found that it's 0.75 parts per billion of chromium-6 in Houston. And telescoping out of Houston, we saw that this problem is really a national problem, and it's very widespread. And so this data produced by the Environmental Working Group tested concentrations of chromium-6 in different drinking water treatment facilities across the nation, and it's at a county-wide resolution. So all of the blocks in yellow are counties where the chromium-6 concentration was below the 0.02 part per billion level recommended by the California Public Health Goal, and everywhere in blue is really the areas of concern. It's above the California Public Health Goal. And so we can see that this is a really widespread problem, and this is really the problem that we're trying to address. As a direct result of chromium-6 contamination, 218 million people every day are consuming chromium-6 contaminated water that's above the California Public Health Goal limit. And so with this problem being so widespread and so prevalent, where do we even begin to address this issue? So Jessica is going to tell you more about our approach.
Now, most drinking water is sourced from surface waters like lakes, rivers, and reservoirs. In the US, surface water makes up 80%, whereas in Houston, it makes up 87%. Since 239 million gallons of wastewater are collected, treated, and returned back into Houston's surface waters every day, we think wastewater makes an excellent target for the preemptive remediation of drinking water. Specifically, wastewater is a great target for chromium-6 remediation, since studies have shown higher levels of chromium-6 contamination in wastewater when compared to drinking water. Now, this over here is an overview of the overall wastewater treatment process split into primary and secondary. In primary treatment, wastewater initially arrives at the plant. It's treated to remove solids and oils. It then proceeds to a bacterial processing system known as secondary treatment. This is where the organic compounds are metabolized, and as a final step, it's disinfected before being released back into the environment. Now, our project focuses on the secondary treatment process because we think it is actually an environment that's conducive for maintaining communities of microbes where we can implement our engineered bacterium into. Specifically, we want to implement an activated sludge secondary treatment process because the activated sludge is where our microbe will live, and it is continuously added to and separated from wastewater in cyclical processes.
Great, now I'll walk us through the genetic design for our project. In this slide, you can see the genetic circuit we've designed in order to reduce chromium-6 to chromium-3 in activated sludge systems. And highlighted in blue here are the biobricks we've submitted this year. We actually submitted 6 biobricks this year from this genetic circuit. And this circuit has 4 main goals, 4 main function goals, which I'll walk through in a little more detail now. The first one is to increase permeability of the cell to chromium-6. And the thinking here is that the more chromium-6 we can get into the cell, the more we can remediate. The next major goal is to be able to sense chromium-6 levels, and this is important because we want our genetic circuit to be active when chromium-6 levels are high, indicating a need for remediation, and inactive when they're low, in order for the cell to appropriately budget its resources. The third goal is really the crux of our project, and that's the remediation goal. And we're achieving this remediation by reducing chromium-6 to its more safe form of chromium-3 by expressing a bacterial reductase. Here we have CRO-R6, which is an enhanced mutant form of CRO-R, which is a native bacterial reductase. The final goal of our genetic circuit is what we're calling a kill switch. We want our bacteria to express a toxin when chromium-6 levels are low. Here we have it expressing BAMH1, which is a type 2 endonuclease. And the idea here is that once the chromium-6 has been remediated, we want the cells to go ahead and kill themselves off. To go into a little more depth on the cell permeability to chromate, here is the system we're actually taking advantage of. It's actually a sulfate transport system. You can see on the right, it's an ATP binding cassette system where the red protein, sulfate binding protein, actually recognizes chromate and sulfate about equally because of their similar oxyanion structure. Also of note here, we chose this specific promoter, PGNTK, because it's a negative feedback membrane stress promoter. We were concerned that expressing this sulfate transport system would increase the burden on the cell, and we wanted to mitigate this with this specific promoter. The next three goals of chromate sensing, remediation, and kill switch are all included in this part of the genetic circuit, so I'll walk through it in this animation. When chromate levels are low, we have constitutive expression of our CRO-B repressor protein, which inhibits our chromate-sensitive promoter, pCRO, so we don't see expression of our reductase or LAC-I. This, in turn, allows our LAC-I repressed kill switch to express our toxin and kill off the cells. However, as soon as chromium-6 is introduced, we have de-repression of our chromate-sensitive promoter, and that allows for expression of our reductase and LAC-I. Then we see remediation by reducing chromium-6 in the environment, and also repression of our kill switch so the cells can survive and continue to remediate. However, as an additional safety measure, at any point we can add IPTG, and this will de-repress our kill switch so that we see expression of toxin again. And one additional design consideration we had, even though we introduced the PGNTK promoter to help reduce the toxicity of the sulfate transport system, we still saw some additional burden on the cells in the early cloning process. So we decided to split our genetic circuit into two plasmids. The topmost portion we've included in a medium copy vector, while the sulfate transport system we've put in a low copy vector.
So far we have tested our system in model organism E. coli, which we are using as a proof of concept, but eventually we would like to transition to soil bacteria Schwannella natensis. We would like to use this bacteria because it has naturally higher metal reduction activity, and it's also natively found in wastewater sites. Therefore, it would have more chances of competing against other bacteria present in those sites, as opposed to E. coli. So in order to assess the change of chromium-6 concentrations in solution, we are using a DPC assay. In this assay, we add a compound called 1,5-diphenylcarbazide to a solution. This compound forms a complex with chromium-6, and this complex has a characteristic magenta color which can be detected by measuring absorbance at 540 nanometers. So we first compared the chromium-6 reduction efficiency between wild-type E. coli and the cells containing sulfate transport system and CRO6 enzyme. From this graph, you can see that the cumulative chromium-6% reduction is actually higher for wild-type E. coli. However, if we look at the percent of chromium-6 reduction normalized over OD, the trend is different. And you can see that for the high chromium-6 concentrations of above 200 micromolar, it looks like actually the cells containing our construct are performing better. However, the error bars in these graphs are quite large. So we would like to also repeat this experiment in the future to confirm or modify this conclusion. So why would we see a different pattern for the percent of chromium-6 cumulatively reduced versus the one normalized over OD? So in this graph, you can see that we compare the growth of the cells containing the sulfate transport system versus wild-type E. coli, we have a much slower growth for the cells containing the sulfate transport system. Since at the zero chromium-6 concentrations, there is basically no difference in growth. We can hypothesize that the The slower growth is caused by the higher import of chromium-6 inside the cell, which causes toxicity and inhibits the cell growth. We also compared chromium-6 reduction efficiencies between two enzymes, CHRO6 and NEM-A. CHRO6 is an enhanced mutant of CHRO-R enzyme found in E. coli, and NEM-A is another native E. coli enzyme. Our experiments show that the two enzymes have similar chromium-6 reduction efficiency. So we also assessed the efficiency of our kill switch induction by EPTG, but we used the fluorescent protein M-cherry in place of the toxin BEM-H1, and we used fluorescence as a proportional cell death response. Our results show that the expected pattern of EPTG induction of the fluorescent protein, and so we had a higher induction levels for higher concentration of IPTG. In the future, we are hoping to characterize the kill switch with the actual toxin in place of the fluorescent protein. Then we would also like to optimize the chromium-6 import inside the cell by decreasing the expression of the sulfate transport system in order to maximize the cumulative chromium-6 reduction. Then we would also test our full genetic circuit assembled as described in the design section, and then we would also like to test our system in Shionella.
So it's now time for the modeling section of our project. This is the secondary treatment system, as Jessica already described earlier, and what we're going to do is implement our bacteria as a part of this secondary treatment system. So there's already this community of bacteria that gets recycled through this loop called activated sludge. And initially, it's added to the water after it has most of the solids, so it's really just water with dissolved organic compounds here. It's added in, it then aerobically can metabolize and grow off of those dissolved solids, and then it gets sedimented out and recycled again so that it can consume more of the dissolved solids. So what we decided to do is implement our bacteria into this loop so it's not only consuming the organic dissolved solids, but also the chromate. And we decided to model this through a simulation on both the intracellular level and the systems level. So on the intracellular level, we have our gene circuit, which is the logic of the whole system of how it responds to chromium and how it responds to IPTG. And we have the cellular growth, which is actually variant depending on whether it's a part of our native bacterial community that's already in the activated sludge that's optimized for it, or our engineered cell population. And then there's also reduction of chromium-6 into chromium-3, modeled on the cellular level. Then, on the entire system level, we did mass balance to make sure that everything all balances okay through modeling of all of our flows. This is where all these processes fit in. We assume that the aeration tank is where pretty much everything exciting happens, which is our competitive growth between the native and engineered bacteria, the chromium reduction, and all other cellular processes, including the gene circuit. In the sedimentation tank, we assume an instantaneous separation of the bacteria in the water. And most importantly, we have these two components that allow for control of the system. We can both add bacteria to control the type of population we have in the aeration tank through bacterial seeding, and also, as it's normally done in activated sludge wastewater treatment, you can purge sludge out of the bottom of the sedimentation tank, because without sludge purging, the bacteria tend to grow in total volume over time. So we wrote some code, and this is actually the core differential equation code that runs our model. And this is the sort of result of that differential equation code. So what happens here is we seed both native bacteria as well as the engineered E. coli that we had, and if you end up just purging to keep the total volume sort of constant over several cycles, what we have is a die-off of our engineered E. coli over time. This is simply because as it can only grow so fast, and it can't grow as fast as the native bacteria. So whenever you purge, you end up taking out the engineered E. coli faster than it can grow. in order to keep the total volume constant. You can solve this problem by doing seeding, and this is actually a seeding regimen that we found works fairly well. This seeding regimen shows that the populations become constant over time, reaching this sort of 24-hour steady state. It follows the standard 24-hour purging process that they always do in typical activated sludge systems. And so whenever you look at the chromium output of the system, you can see that after a few days of implementing our system, it is well below the California public health goal for drinking water. Through our modeling, we were able to find that if you were to implement this, you need to do bacterial seeding, and you also need to do, or you also can get the amount of chromium within the maximum contaminant level.
Finally, I'll be taking the high-level science and modeling concepts and bringing them back down to earth to Houston and explaining the human practices activities our project did this year. Initially, we conducted extensive exploratory research to learn more about the severity of the problem in Houston. Now, Houston, we have a problem. People know about arsenic, lead, and other contaminants, but they don't know about chromium. Also, the current regulations in Houston don't maintain levels of chromium that are low enough to ensure the safety and security of our citizens. From these realizations, we developed these goals of first, raising awareness of chromium-6 in the community, second, influencing the regulations revolving chromium regulation, and lastly, proposing our synthetic biology solution as a way to remediate the current chromium concentrations in Houston. We strove to achieve these goals through numerous ways. First, we screened the movie Ellen Brockovich, a film about chromium contamination lawsuit in California. We also presented our project at many luncheons, symposiums, and conferences to share our knowledge with the scientific community. And lastly, we hosted our annual bioethics debate where we invited high school students and asked them challenging questions about the ethics involved, the ethics of synthetic biology, and made them excited about the field. On a side note, we continued our project from last year, the Humans of SynBio, where we gathered stories from scientists in the SynBio community all around the world and shared it online through Facebook, Twitter, and Instagram to address the negative stigma held by many people in the public about the field. As a capstone of our project, we visited the Baker Institute for Public Policy and met with Dr. Kristen Matthews, who mentored us in drafting a policy brief that we plan to send to the Houston City Council about the current regulation of chromium-6 and about implementing our project in the wastewater systems. In future works, we hope to integrate the modeling and human practices and conduct an analysis of the costs related to maintaining the viability of our bacteria in the wastewater treatment system, considering factors such as seeding and others. These are a few of the teams that we collaborated with this year. The University of Valencia and William & Mary were people we interviewed for the Human System Bio. We also met up with all the other Texas iGEM teams in a conference where we shared ideas about synthetic biology and presented our projects. As an overview of our project, we would like to point out a few of the things we're most proud of, such as our modeling, collaboration with UT, our Trans-Texas iGEM conference, and our integrated human practices and modeling. In conclusion, we'd like to acknowledge our PIs, advisors, and sponsors. We're so happy and grateful to be able to attend this Jamboree conference, and we would not have made it here without them. That concludes our presentation. Thank you for your attention, and we'll accept questions.
Questions from the
Great, creative and sincere job. Thank you for the presentations. Lac promoter is a leak promoter. It's weak, regulated by lacky I repressor. Did you thought about it? Because maybe it can cause impact in the cell machinery before the bioremediation occurs. The Q switch.
Yeah, that's a great question. And I think that probably is something that we would continue to refine in the future. But for now, it's mitigated by the fact that we're reseeding our bacteria every 24 hours. So if more bacteria are killed off than we intended, that would sort of help in that respect. And also, our project on the whole is erring on the side of biosafety more than anything. So I think when you're confronting public that's generally skeptical of GMOs, if more of our bacteria are dying off than we intended, it's not necessarily a problem in our eyes.
But it's more expensive. It has promoter well regulated than the lucky promoter. And a suggestion, your circuit design was really good. It's well founded in bioengineering principles. But it would be more clear if it was in Boolean logic gates, do you know? By ND logic gate or not logic gate. It's huge and it's beautiful. It would be more clear. But it's really good, guys.
I have a question about the waste blocks. So could you show that slides? You explained in a certain part to add the bacteria for the bioremediation. Yes, that one. And then in that second tank, you get rid of the bacteria by the sedimentation. Did you study this process? Is that just a way to get rid of your bacteria?
So the reason it's sedimented here is because in wastewater treatment you want to recycle the bacteria. So you want much more bacteria than you could produce off of the metabolic amount of biochemical oxygen demand or just dissolved organic compounds that could flow through at any one time. So the reason you have this recycling process is because without a recycling process, you would not be able to maintain as large of a community of bacteria. So it's...
And is this sedimentation about 95-100%?
Yes, it's almost completely efficient. Also, if any bacteria do slip through sedimentation, they're pretty much immediately killed by UV light in the disinfection stage, in addition to our other kill switch design components.
Just one quick observation, very good presentation, where you compared the natural or native enzyme, the name A. Can you go to that slide quickly? And yes, no, the other slide.
Oh.
Next, forward, yes, this one. Do you notice, can you interpret that in another way than saying that the MG compared well with the name? Is there any other thing you see in that slide, especially at higher chromium levels? Do you notice a striking change that at higher chromium levels, you're comparing the native enzyme and the one you cloned. So, did you see a switch? At lower chromium levels, the MG1655, does it do better? A reduction. Is that the slide I was looking for?
So
and you see a switch, a transition.
Here we looked at the reduction percent chromium reduction normalized over OD, and the trend that we noticed that for the concentrations above 200 micromolar, there is yes a switch in a trend, and we see the system containing our circuit exhibiting higher chromium reduction activity compared to wild type E. coli.
Here's the next slide also. I didn't want you to miss that. There's another slide. Go ahead. Where you compared, yes, this is the slide. You notice something there? So the conclusion you gave us in your presentation was that they were both the same. But I see a clear trend. Do you?
Well, so for the 50 micromolar, we have corrosives having higher chromium reduction activity, but then for the higher chromium concentrations, the NIM-A is reducing slightly larger. But again, since we have like a very large error bars for CRO6 enzyme, we would like to also confirm this with further experiments. But yes, this is kind of a trend we would like to explore in the future.
Okay, thank you, and we'll follow up at the poster session. Thank you all three teams for presenting today, and now it's time to enjoy lunch.